It doesn't say.
Oh, you have to get out of it.
Oh, it's no,
Okay.
it's okay.
It's okay.
Mm.
Because people is complaining that they cannot
Okay.
hear.
So is that okay now? Okay. So I'll start my presentation. So thank you for having me here. So it's great honor to be here because I heard about this computer center a lot from Japanese colleagues and collaborators. And I was wondering how it looks like, and kind of it's really huge, and I'm surprised and amazed that so many people are working for life science using a supercomputer. It's really, really nice to hear that. So our laboratory been working on the signal network, particularly for the signal network. And I'm originally biochemist, but I started to realize that only experiments cannot solve the question we have for the cancer signal network, because you know that the data is sparse, and the data we take, we say it's a lot of data, but it's not that a lot of data for computer science. And the prediction is very difficult using only experimental data. So I started to use develop the mathematical model to understand the mechanism of the signal network, which is very important for the human diseases, particularly for the cancer. So I will talk about the cancer signaling network and our recent approaches to use the natural language processing to develop mathematical model. So this is the cancer network we are dealing with. So it's kind of very old papers, but it's well explain how that signal network affect the cancer growth or cell death. So that about 15 signal networks operated to define the cancer cell growth. And the most difficult part of this cancer signal network is the input and this kind of output. A relationship is very non-linear because there is a lot of crosstalk between the pathways, and there is a feedback regulation everywhere. So because of this complexity of the network, it's, as I said, it's very difficult to predict the drug efficacy only using the experimental data. So actually those are that the drugs are developed to target the signaling network. You see that many cancer drugs and already many other drugs coming and waiting to be developed. And sometimes because of the side effects or drug resistance, such a development have to be stopped. So we want to have a larger high success rate in drug development. And to understand this kind of problem of the drug resistance and the side effect before the clinical trial, because the clinical trial is very expensive and takes time, so that the prediction before the clinical trial is very important these days. And particularly even in the Japanese pharma, they want to develop the predictive model using a limited amount of experimental data or clinical data we already have. So that for that, that the mechanistic modeling and modeling using AI and so on are very important, particularly for these 5 years. So that today I will talk about the mechanistic modeling of the cancer signaling, and I will talk about example of the patient-specific modeling using such a modeling mathematical model first, aimed for the basic understanding of the cell fate control. And the second, I will talk about natural language processing to make a context-dependent model. Context-dependent means that the disease-specific or cell type-specific, whatever. And in the third topic, I will talk about the multi-scale simulation it means that the combination of the mechanistic modeling I'm explaining here, and also the molecular dynamics simulation at the protein level. So this is the network. I cannot see. Sorry, I don't know how to stop this. But anyway, so this is the cancer signal network we often use as our toy model. So that this is called ErbB receptor signaling. It's also include EGFR. And this ErbB signal receptor signaling is really problematic in the many types of cancers. And but this receptor has a very specific features. Like receptor family has 4 receptors, but they make a homodimer or heterodimer, and they bind to the different kind of ligands. And you see a lot of combination here, but in a cytosolic pathway, it's kind of still there is a overlapping. The different molecules which can bind to the different receptor combinations, but this part of the kinase activation module is kind of very much limited. So it shows this kind of bow tie structure, and then they emerge to the activation of the transcription factors. Then they go to the sulfate control, so that this part is like 60 minutes when we measure by the experiments. And this transcription factor is like a wavy, like things like a first wave and the second wave and the third wave, and it's like 1 day or something. Then they go to the cell cycle entry after within 24 hours in our system. Then that cell fate decision is done by the 2 or 3 days. So we often use this ligand, EGF and heterogeneity, which bind to the different types of the receptor. And what we focus is the difference of the dynamics. So that when we look at this ARC kinase, then the EGF shows a transient, and heterogeneity shows a sustained, and then they show the completely different activation profile at the C-force activation. So that even though the EGF shows that this transient activity, but at the level of the C-force protein activation, it's nothing. But heterogeneity shows a full activation. And then they go to the differentiation and the proliferation again, so the different phenotype. So that the first information is analogous, but at the phase of the C-force after 60 minutes or something, they go to digital. So that and then the cell fate is also digitally determined. So we make this kind of model, describe this the process by the detailed protein-protein interaction and kinase or phosphatase enzyme reaction. So this is pretty much complicated, but it shows the arc to the C-force protein expression and activation. And we usually take this kind of time course data in our laboratory. In this case, this 1 goes to this measurement, and the 2 goes to this measurement. And we do the parameter fitting of this model to that experimental data, and define the parameters, and do the several kind of validation experiments in our lab if the network is trustable or not. And then we go for the sensitivity analysis, it's called. We like to use the sensitivity analysis, and because it often can identify the drug target. So the sensitivity analysis is changing to change the parameters of that each reaction, and then see the output, in this case, the C-FOS mRNA and the C-FOS protein. We can change the output depending on the purpose, but we look at the bottom reaction of the network. And from this network, we found that the DISP, which is important for the ARC, the activation, is showing the highest sensitivity. So it means that the manipulation of the DISP is effective to manipulate the C-FOS output. So we think this sensitivity analysis, interpret the sensitivity analysis results in this way, and also validate by the experiments. So that network, which looked very complicated, can be explained by this simple network. It shows it tells like a double coefficient feed-forward loop, naming in this inner and outer loop. And in addition to the coefficient feed-forward loop, we have our AND gate, the C-FOS, and also the DISP, and also the negative feedback regulator from the C-FOS to the activated C-FOS to the C-FOS expression. So in this analysis, we focus on the role of this the double coherent feed-forward loop, and we performed some analysis with Boris Korodenko, a collaborator who now is in Dublin, and then we checked we added the sine wave of the arc and see that how this C-FOS output has been changed with and without inner loop. And we found that with the inner loop, that the C-FOS didn't go up so dramatically. It means that the inner loop is, keep the system more robustly. So that without the inner loop, it goes up so quickly. So that we see that this the transcriptional network to control that the robustness of the transcription activity is important in this system. And we further analyzed the effect of the negative feedback, particularly for the DISP. And the DISP is described by this parameter K3. And then we perform the robustness analysis, changing this K3 constant. And when we see that increasing this constant, we see that not much, the upregulation to the C-FOS. So that again, we see that this negative feedback also contribute to the this ARC to C-FOS network. And once we developed such a complicated model by writing many ODE equations, we want to see that how such a model, we spend a lot of years, can be analyzed for another cell line or another system. And then in this case, we first made a model using MCS breast cancer cells, but later we also checked with the PC12, which is a standard cell line used to study the glioblastoma. And then we found that this different cell lines shows a different, even though those cell lines are stimulated with the different conditions, but it shows a very similar profile after the simulation and the experiment. It means that those cell lines are operated in the same common principle, which I showed in the slide. So now that was a kind of example we basically run in our laboratory, but we also have a question. Even though we studied such a cell system using a cancer cell line, but in a real world, a lot of variation in a cancer patient, and also in the data. So this is an example of the breast cancer subtypes. So that the breast cancer subtypes contains 4, like a luminal A, B, and HER2, and basal-like, it's called the triple negative breast cancer. And usually the standard therapy for the breast cancers are based on this only 4 subtypes. And particularly for the TNBC, red one, they don't have any molecular targeted drugs. So, and also there is a problem for that. But even though if we look at this luminal A, B, HER2 positive, they are standard therapy. But if you look at the individual patient, you see a lot of changes in mutations and copy number variation. So this may cause drug resistance to each patient. So that, and in particularly, those changes are really, really enriched in a signaling pathway. If you look at those genes, most of the genes belong to the signaling network components. So those study, the cancer genomics focusing on the mutations of this, each subtypes, but they often use the statistics. But the combination of those all genes cannot be analyzed using a statistical method. So that and I believe that the ODE model can integrate those parameter changes and initial value changes explained by these mutations and the copy number variation can be used to make a patient-specific model and analyze the behavior as the dynamics of that system. So but there is a challenges, for the patient-specific modeling. So this dynamics can be explained by the network information and the parameters and initial values. And the network can be, constructed from the literatures and the databases, and the initial value can be obtained from the transcriptome or proteomics these days, even from the public data. But for the parameters like a K-on or K-off, such things cannot be obtained from the patient data. So that this is a very difficult point to make a patient-specific model. So our laboratory decided to use the parameters obtained from the cell lines. So we used the 4 different subtypes of the 4 cell lines which correspond to each subtypes, and they performed the parameter estimation using the data obtained from this. And we also used the initial value of the transcriptome data of this model, which contained 34 genes, and we used the transcriptome expression data of this 34 genes of this cell type model, and then they estimate the parameters. And in this case, we try to get the common parameters can explain the 4 different cell lines. And then using those, the cell line parameter, we use the initial value of the patient as using a transcriptome data from the TCGA. So that the cell-specific model, we use the cell line data initial value, but here instead, we use the cancer data, but the parameter is the same. And then they perform the simulation for each patient, and then they get those dynamics. And this is the results of the one of a few example of the data, how it looks like. So that we performed the 377 breast cancer patient data and perform the simulation of this AKT and ARC and MIC as an output, and obtain those in silico dynamics. And then we analyze, at the time, we analyze this max value or drop rate or AUC of the dynamics. And then stratify the patient using those database, those dynamics. So that here is a result. So we see that this red color, this is a TNBC, has a poor prognosis group and the better prognosis group. And we found that this poor prognosis group shows a lower heterogeneity in silico heterogeneity response. And we see that this is because that this poor prognosis group has contained a higher EGFR expression ratio compared to the other family receptor members. And this may be a reason of this dynamics changes. And then we, again, that use the sensitivity analysis for each patient group. And then we found that the lower EGFR sensitivity is associated with the poor prognosis. And then we again analyze the public data to validate this hypothesis. And indeed, that the EGFR inhibitor showed that not responded very well to this prognosis group. So that was a basic pipeline we used to develop the patient-specific model. And at the time, it was before 2022. And then after that, the AI and the deep learning is, can be commonly used. So we, this guy that Masato has developed the self-supervised, non-constructive and label-free learning of the one-dimensional neural network method. It's called the DIM profiler. And this is, to encode all simulated dynamics obtained from the simulator, so that including not ARC or AKT, but we can get the dynamics of the all intermediate, the activity as well. And then and also using this method, we can identify what is the reason of the classification. And we did this analysis for each year of the prognosis, and we found that this DIM profiler is more accurately predict the 5-year or 7 years patient prognosis. And we could find another biomarker, GSK3 beta, for the of the reason of this classification. So so now we kind of set up this kind of patient-specific model, and we called it the small pipeline as a Pasmopy. And this Pasmopy can do the parameter fitting, critical data input, and the simulation analysis. And we also actually used this the text model to develop this model. And so that this is a kind of reason we moved forward to the natural language processing. So the text model is a method. We are using pattern matching of this biochemical reactions and equations. So at that time, we didn't use any, nice AI and so on. This is very simple pattern matching. But I think this is nicer now, because now we use a lot of NLP, but this pattern matching is very simple. We can register this biochemical words like binds, phosphorylation, or transcription, and then we can generate like a mass action or make arrangements and reactions. It looks simple, but you can add more this event to this text model. And also after that, I found that the biological data is very noisy. No, I mean the biological description of the reaction is very noisy. Not many people write like A binds to B. People write in more literature like, prefer literature like writing. It's not like a student writing. So that this text model can be used at the intermediate language to concentrate such a noisy biological text to the more simplified word, and then go to the modeling. And actually, we developed the model using this text model, and then they performed that analysis what I showed. So we now text the model, and we also had the pipeline, which is called the biomass. And the biomass can do the parameter estimation and also the sensitivity analysis without the need of the skilled knowledge. And we can combine text model and the biomass. Then you can perform the simulation as a minimum knowledge of the programming and the minimum knowledge of the mathematics. But again, now we have a problem because to make a text model, then you need to know the gene-gene interaction before writing the text model, A and B interaction. So that now, then we went to the natural language processing because we spent so much time for reading papers, like to construct one model. We did hundreds of papers. If we want to develop the breast cancer model, we need to look for such papers. So it's really, really time consuming. So we used we first tested like a structured database, like a KEGG, to make such a model from KEGG, because the KEGG is just a map, not the dynamic model. So that we used the KEGG for that, and also data. And now we are spending so much time to extract those knowledge from the PubMed. So that I will explain this one today a little bit. So that we think that the context dependency is very important for us. And look at the metabolic network showing here. So that the bacteria, yeast, or human all has a metabolic network. But depending on that, the yeast name, and in the case of human, like a diabetes patient may have a different metabolic network than we have that the normal healthy people have. So that we need to have a context-dependent network. So that to do so, right now we are using a BART model. So the BART model was first trained with Wikipedia or Google Corpus, and this is explained like this by their literature. So that if that doc is explained, it contains the information of this like acute or praying and so on. So the doc has several meaning by writing doc. And it's same for genes and proteins. If say we A gene or B genes, there is also such a similar information, associated with this A or B genes. So we want to have those information because we've been, when I was in weekend, before I moving to Osaka, we are struggling with the data integration for years and years. So that the problem I see from now that, A gene or B, A protein and C metabolite, something like that, so that we just looking at the pattern matching, but this is far beyond the pattern matching, so that I think we can integrate the biological data nicely, much nicer than the pattern matching method. And I think that is very necessary. And even though many people start to use those methods for the data integration or classification, and I think that we can do it better if, like 5 years or 10 years later, because we have those nice methods. So this is our collaborators' paper now appeared in the archive, so that they tested more than 1,000 language models, and then they see that that each developer has a very close approximately of the characters in language models like that. But what we concerned is how the language models are trained. So that this is an example of the PubMed and the archive, and then they are close, but the model which are trained with like a Wikipedia are very different. So that it means that that when we use or utilize a language model, we need a language model trained with biological information. So that in this case, PubMed, but we can train with the experimental data or the other database and so on, so that that can be definitely possible. So that this is first confirmation that such a context-dependent extraction it can be better than the normal PubMed things. So that we use the information of a query using a mesh term. Mesh term is associated with the different human diseases, and mesh term tagged or not tagged with mesh term. And made ask the query. In this case, we ask the query of the these 6 queries, and that we tag the same this tag as a mesh term. And then we see that this breast cancer is more enriched with the mesh tagged 1. And even though it's breast cancer and triple negative breast cancer is same, but supposed to be in the same category, but the TNBC showed a higher accuracy in mesh term tagged query. So that this is what we saw. So that by using this, we have like a context dependent vector by analyzing vectors for each and get the information of the network. And also we see we can use like co-occurrence type analysis using PubData 3, which is already labeled with the like a cell line or gene name or the other functions. And then we combine this information together. Then we can get those network specific for the breast cancer and the type 2 diabetes. So that this is a very simple the model, but we see that the AKT or TP53, which are very common for the breast cancer, are highly enriched, and also the ErbB2 is here. And for the insulin, we could get the insulin or AKT also, which is supposed to be important for the insulin diseases. So that we can specify more, the asking, changing the query and so on, so that this will be possible. And now but our final goal is not, extracting network, but we also go for the mathematical modeling. So we want to use our data together with such literature information. So we we have our differentially expressed genes of the breast cancer in our laboratory, and then we use such a gene expression deck as a node of the network. And also we used such a literature-based network and combined to network node. And we finally, and also we used the biomodels node. And then we could get those network, in a data-driven manner. So this is ErbB receptor again, which we used in the earlier analysis. And this network is fully manually created, but we see that those interaction and the network at around here is well captured by this method. So and now we want to go for the executable model from those things, but also we want to make sure that this network structure of this this data-driven model is feasible to the already known biological network structure and so on. And we need to work a little bit more. And this is what we are doing. And I think this morning I had a really good discussion with the Mariko and the the other people. And I think there is a lot of demand for this kind of NLP-based network instruction and the application of this network and the mathematical model for the human disease analysis. And I also wanted to do this kind of multi-scale simulation I showed here. So what we are doing is a mathematical modeling, but the mathematical model is made of the protein-protein interaction and the kinase activation. And as shown here and earlier, those proteins and the enzymes are very much mutated in cancer. So such a mutation can be, affect those network output. For example, EGFR has many mutations in lung cancer, and they may change the association with the other proteins in the network, and they may affect this one, go for the cell cycle and so on. So that we can estimate this, K on and K off by the parameter fitting, even though we need a lot of data to make an accurate parameter from the data, but technically it is possible. And for the MD simulation, we get those structure from the PDB database, and we can calculate this the association by the free energy changes, comparing the wild type and the mutation. So by doing the parameter estimation from this side and this side, we may find that mutual the normalization method, how to incorporate the MD data to the mathematical model. And also we may able to find the alternative pathway which cannot be explained they only considering this information. So this is really preliminary, but this is like a sub-model, just focusing on the EGFR. Then the EGFR make a dimer and phosphorylated, and the phosphorylated things bind to the c-Myc, one of the target protein we are looking at, and then the c-Myc associate and dissociate and make this the complex. And this can be explained by this ODE models, and we can compare with this too. So that this is the data we obtained in our laboratory. We focus on the 5 mutations well known for the lung cancer mutations, and we also got this kind of the kinase activity from each cell line by the Western blot. But now we are thinking of the using a proteomics and other method. But so far, proteomics Western blot is more quantitative than the standard proteomics so far. So that for this, we are using the Western blot. So that there is a slight difference, not much different, but there is a difference, and also the output is also different. So we calculated this large KD by taking a ratio of the K1 and K-off, and we see that this, because we performed many parameter estimations, so there is a variation in the parameters, and but still we see that there is a which is higher and which is lower. But that is what do for the cell simulation, and for the molecular dynamics simulation, we are collaborating this project with Dr. Ai Shinobu in Osaka University, and she can access to the FUGAC, so that we use the FUGAC supercomputer in Japan, and we use the replica method to run the simulation in a parallel manner. And then to estimate this free energy changes, we use the MMPBSA method right now. But because it showed a good trade-off between accuracy and speed. But I heard from Shinobu that nowadays there's a more, having they started to have a good theory to predict the K-on and the K-off, not the large KD, so that we are trying to test such a method once the correct things are done. So that this is a preliminary result we obtained by comparing the MD and the mass mechanistic simulation, and we see that there is not that good correlation, but we also see that there is in the case of the free energy calculation, we see that those mutation may have a lower affinity to the SIC, and this mutation has a higher affinity to SIC. So now we try to define the method of the free energy calculation, and also for the parameter estimation, we are trying to get more data to have a better prediction of the parameters in a mechanistic model. And what we want to do is, now we are working as a model, working this kind of model, but we also do that similar thing to the MD, so that the thinking about the dimerization and the phosphorylation changes, SIC binding and other receptor binding, and also consider the alternative pathway to the model. So to do so, we are testing the other MD. We're using enhanced sampling. So this method, we see that the wild type showed active and inactive states without ligand, but this mutation has an active state, only active state, and this mutation has two states and so on. And we think that there is a structure which called ErbB and EGFR. They may affect the SIC binding, so that the mutation near the ErbB or the far from the ErbB may decide the affinity to the SIC. So we are now testing this hypothesis. So that here is my overall summary. So that we've been working on the mechanistic modeling and for the patient-specific model. And then we started to use the natural language processing. And I think I need to work more on this parameter estimation and the network identifiability for the large scale model, because the NLP based model can be large, I think. And we want to get such a more solid results to remove the uncertainty of the model. And also, we want to combine the other omics data from the mathematical, and combined with the mathematical model by the data embedding and so on. So that this is kind of our future direction, or what I want to do. Actually, we are getting a funding from this kind of a project, so that now we are doing the literature and the embedding as a math model so far. But we want to combine this omics data and analyze the whole network. And it's called like analogy task. We can test the similarity, or the simple calculation of the A and B can be explained, if possible, can be explained by like a bind or phosphorylate to analyze the interaction and combine those information, so that it will be done. I think it's very nice, and long-term biological question can be solved somehow. So this is my laboratory members. So this text model was done by the Hiroaki Imoto. Now he is in the University College Dublin with Boris Korodenko. And the NLP modeling is done by the Kiwamu and Masato. But we also get many experimental data from those people, and we collaborate with the other people, like data embedding and MD simulation and so on. And thank you for the funding for this inviting to me here too. So thank you very much for your attention.
If no one has a question, I will start with one. So one main problem that we found out by working using automatic process to creating network is indeed the noise, the amount of noise that is in the prior knowledge. That comes from the fact that many interactions are annotated in literature, like with opposite signs sometimes, even from the same context. And this could be like maybe the quality of the data or differences in the practical experience. So do you think that by filtering so much the context, we can have networks that are consistent?
Yeah. Well, actually, we also worked, we had a similar problem with this too. And we tried many things on that too. And we ended up to analyze the DEG transcriptome data. And but the idea is, if there is a correlation of these two genes in transcriptome data, that such an interaction is really happening in a cell. But at the end, we found that such a correlation is only 10%. So that now we believe that the proteomics data is better, because the proteomics and the transcriptome correlation is not nice, and many genomic studies revealed that. But in terms of particularly we focusing on the c-Myc network, we know that which is upregulated than which is supposed to be upregulated, and we can tell it's not 100%. I believe, because I see that the like a peak of this maybe it's better. So that even though it's the same network, for example, but arc appears like 5 minutes, but this the C-force it appeared 90 minutes. So that when you think about this 5 minutes correlation with this and this, the correlation is very low, but still I believe this is better than transcriptome. So that now I'm thinking to use the proteomics data to eliminate the noise. And also, if we have a lot of money, we need to have such a consortium to modeling-based proteomics project. Then I think we can solve the many problems on the cancer, because if you block the network by the anti-cancer drug, you also see the different correlation, and then we can use that for the removing the noises, or to see the difference of the network between. I think that is important, and I totally agree about your problem.
Questions from the audience? No, so maybe we can ask just one more question, sir, about the coupling between molecular dynamics and ODEs and ODEs system. So what concerned us when we discussed with how we could couple Boolean networks and molecular dynamics simulation was the differences in time scales. Because if I understood correctly, molecular dynamics simulations usually simulate in a span of femtoseconds, if I'm correctly.
Well,
While
nano,
instead,
nano.
where the kind of simulation we do, they are more in the minutes. So there are like so many order of magnitude of differences. Is it something that worries you when you do this? Or?
Yeah, maybe it's also important, but I'm rather the care about the stable things like a protein-protein interaction, the difference between before and after the binding, and how the mutation affect the this the state, stable state. That is so that I may use this MD simulation, not using full power of the MD simulation, but right now my concern is to look at the protein-protein interaction, and to see simply to see the steady state of the mutation with and without mutations, and if there is changes in the state. And actually Shinobu and I did several literature searches about this EGFR mutations, but not many people are actually studying it. So that they use this mutated protein as a the docking study or a drug things, but they don't do this in this way. So that I think as a basic research, there is still things to do.
Thank you.
Thank you.
I mean, on this combination, that's the time for the talk. I mean, you cover many of the things we are interested in. All the things we are interested in.
Thank you.
The problem with molecular dynamics is that it's very expensive. So
Yes.
for all proteins,
Yeah.
mutants, all something. So we need something lighter.
Yeah.
Machine learning design systems or something.
Yeah, and also we, try to use AlphaFold as much as possible. Yeah. Reduce the hypothesis. I mean that many possible hypotheses to make a small number of the hypotheses, and then and the good thing about the collaboration with Shinobu is she can access to Fugaku as a member of those, and also their they have once she was belonging to some group in Riken, and whose name is Sugita. Sorry. And Sugita's group has developed many methods, including it's called a replica method, so that they make a copy of many, the types and learn. And also they do the, I forgot the correct name, but, the more, the crude way of the, molecule, the atoms in MT simulation, so that they can combine such a low-cost simulation as well.
Now we have there is another group in the department that works in metadynamics. That is it's much faster than molecular dynamics and something similar. Then for how to get from the literature the Boolean networks, how to reduce the number of errors. I think at then it's very difficult to, if you just go for single sentences, is this bound to that, or is this something they will be able to contradictory information? So I think we need a better way of evaluating the quality of the papers. Is this a paper that contains many different experiments showing that? Normally a paper is like 5 different experiments to demonstrate that this is targeting that. So something that will go more than just finding a single sentence or single
So
attached to this, how many experiments has been demonstrated.
It's true.
There are already systems doing that. For ErbB Journal, they developed a very nice system for extracting hypotheses from the papers based on what is the conclusion in the figures of the different experiments. So the papers from this first figure, they demonstrate that in second line, and then give them a full hypothesis for the paper. So they will be better than just stopping similar parts from the other. Oh, I think it's a very interesting topic.
Thank you. Actually, as you said, finally that they had a benchmarking and, the validation of this text information is pretty much different compared to the normal omics analysis. Omics analysis has a lot of background data, rather easy, but this kind of description is very difficult. So that is why we stick to the signaling network, which particularly for this ErbB signaling network, because we know better. So that and then once we develop the method, we can apply for the other, system. That is our strategy.
It's very important to have a good validation.
And also good thing about ourselves, it's not that difficult to experiment. We can do by ourselves.
No more questions?
One question. You mentioned in the presentation that at some point using statistics was not useful because mutation. Am I right?
Could you repeat again?
Yeah. At some point in the presentation, you mentioned that using statistics was not very useful because mutations.
No. I saying that statistics is very important to obtain the this one, right? So for example, what I wanted to say is that to include statistics is very difficult to handle, to include all those mutation data as a variable to classify the patient, because combination of the two is already too much for the statistical, because to identify this cancer specific susceptibility gene, you may need like a 500 patient data for identify the, statistically significant, the disease genes from the, noisy, normal people have the, the mutation and somehow. So that it means that if you consider the two mutations combination, you need 500,000 births at same 500,000. It's impossible to collect such a patient data. So I mean it in that way. And also the more than 2 is also statistical, statistically, I think it's also difficult because of the combinatorial, calculation. That is why. Okay.
It just have like a very, very last question. First of all, amazing the talk and how many scales are available to integrate. And my question is more like kind of philosophical, because you let's say model systems represented as networks, and you try to understand the dynamics. Okay, for instance, you have cell differentiation, where you not only have cells that respond dynamically to some inputs, but it's also the network itself, like changing along time. So I was wondering if you foresee that this type of methods could be extended to phase this type of transition. No? Because nowadays we would like to work, for instance, with organoids, organoids, and we really want to have like good representation that will evolve over time.
Actually, we are working on the cellular senescence, and it's collaboration with a pharmaceutical company with the skin aging, so that they want to develop the good cosmetics for the skin age. So that the problem is that aging is takes time. So that I think that there is really complicated this kind of feedback and AND gate and feed forward. So that I think first I believe that the network will be changing, but also that the network may be same, but at some time the parameter can be changed over the time. Network may be same, but I don't know how to make this things. But I think for this kind of network, and also the same for the drug treatment. If you get the drug at some point, and after several years, you may have a relapse, and what changes in the network. So that I think it will be very interesting. So that I think right now we are just focusing on very short time, but I think network the cellular senescence and the aging, it's kind of challenging project for me. But I say that and also aging is affected by the stiffness, the mechanical stress. This is our recent finding. Sorry, there's a lot of female scientists, so I will tell you. So that softness of the skin is important for the use of your skin. So that the skin get higher, then the cell get aged. So that is our the recent findings. So that mechanical stress, you think that it's not related to aging, but actually it's related. So that I think those things are pretty much what we thought very different, but it's connected in the long term. Yeah. Thank you.
Thank you very much.
Thank you.